Hello, let's discuss on the topic meiosis. Meiosis is a type of cell division in which the chromosome number of the resulting offspring is reduced to half as compared to that of the parent. Therefore, meiosis is also called as reductional division, and this meiosis occurs in the gametes or germ cell. Now, meiosis follow two divisions, that is meiosis 1 and meiosis 2. In meiosis 1, the chromosome number of the parent is reduced into half. So in meiosis 1, two individual arise from a parent cell. Now here in meiosis 1, the chromosome number of the parent is reduced to half. That means the two resulting offspring contain half the number of chromosome to that of the parent. Let's take an example. In human we have 46 number of chromosome, and this 46 number of chromosome is divided into two sets of chromosome. That is one set containing 23 and other containing 23 sets of chromosome. That means two set, or we can also call as 23 pair. So human contain 46 chromosome or 23 pair of chromosomes. Now this 23, one is called the first, it's called this 23 is forming one set, the other 23 is forming another set. So there are two set. Therefore 2n refers to 2 means 1, 2, n refers to the number of chromosomes occurring. So 23, 23. Therefore 1, 2, when we multiply with 23, it is giving rise to 46. So this in human, this is containing 46 number of chromosomes. Now diploid means what? Di means 2, ploid means set. So 1, 2, there are two sets in human. The chromosomes contain two sets. In human there are two sets of chromosomes. Therefore we human are also called as diploid organisms. Now in 2n we have 46 number of chromosomes, and during meiosis, as I told you, that the number of chromosome is reduced to half, and each of the resulting offspring will contain half the number of chromosome as compared to the parent. So half means 23. This also will contain 23. So here the number of chromosomes is reduced to half when we compare with the parent. Therefore this meiosis 1 is also called as reductional division. And in meiosis 2, the chromosome number of the resulting offspring remain the same. That means this is the original chromosome, and from this is the original cell containing 23 chromosomes. So the resulting cells from this is containing the same number of chromosomes. So here n, parent is also n, this is also n, n. So they will all contain 23 number of chromosomes. Now therefore this meiosis 2 is also called as equational division, because in this case the offspring are getting the same number of chromosomes as like that of the original cell. Therefore in meiosis the resulting offspring will contain half the number of chromosomes as compared to the parent, and at the end of the meiosis, 4 daughter nuclei or 4 daughter cells containing haploid number of chromosome or half the number of chromosome develop from this meiosis. Meiosis is there are two divisions in meiosis. The first one is meiosis 1 and second is meiosis 2. Meiosis 1 contains 4 phases, that is prophase 1, metaphase 1, anaphase 1 and telophase 1. Now under this, let's discuss on prophase 1. Prophase of the first meiotic division, that is meiosis 1, is typically longer and more complex as compared to the prophase of the mitosis. The prophase 1 is further divided into 5 subphases. The first subphase is the leptotin. Lepto means thin, tin means thread. So leptotin means thin thread. Now during this leptotin phase, the nuclear membrane remain visible and intact, and the chromosomes may attach to the surface of the nuclear membrane or remain free inside the nucleus. And each of the chromosomes at regular intervals have a bit like structure, and this bead arises because of the condensation of the chromatin fibers. So these are the basic characteristics of the leptotin phase. Second is zygotin. In the zygotin phase, the nuclear membrane also remain intact and visible, and each of the homologous chromosomes, there is a pairing of homologous chromosome. Homo means same, so there is a pairing of same or similar chromosomes. So this pairing of homologous chromosome is called as synapsis, and the paired chromosomes now are referred to as dyad or bivalent. Dyad means 2, bivalent means 2. So the paired chromosomes are now referred to as dyad or bivalent. The third phase, subphase, is the Picatin. In Picatin, the bivalent chromatids divide, and it gives rise to 4 chromatids again. So 1, 2, 3, 4. So there is a separation of the bivalent chromatids to give rise to 4 sister chromatids. So these and these are the sister chromatids, these and these are the sister chromatids, but these and these are non-sister chromatids because they are belonging to different chromosomes, though they are homologous in nature. And there is also crossing over occurrence between 2 non-sister chromatids. What is crossing over? Crossing over is referring to the exchange of genetic material between 2 non-sister chromatids. Now, the point of crossing over, that means the point of attachment between 2 non-sister chromatids, is referred to as chiasmata. And due to this crossing over, the exchange of genetic material between 2 non-sister chromatids arises. Now, the fourth subphase is the diplotin. In diplotin, the bivalent begin to separate from each other, except at the site of the chiasmata. The chiasmata is the point of attachment is the site where the 2 non-sister chromatids attach. So there is a separation between the two. There is a tendency of separation between the 2 non-sister chromatids, but the point of chiasmata remain attached still. The fifth stage and the last stage of the prophase 1 is the diakinesis. The diakinesis is the final stage of the meiotic prophase 1. This phase is marked by the terminalization of the chiasmata. Terminalization here refers to separation of the chiasmata. And during this phase, the chromosomes are fully condensed and the meiotic spindles begin to assemble so as to prepare the homologous chromosome for separations. So spindle fiber begin to reappear in this diakinesis, and this shaded portion indicates the recombinant genetic material because of the crossing over between the 2 non-sister chromatids. Now with this, prophase 1 completes. Now the second is the metaphase 1. In the metaphase 1, what happens is that this metaphase 1 and the following till telophase and cytokinesis is similar to that of the mitosis. In the metaphase 1, the bivalent chromosomes, they begin to assemble and align at the equatorial position, forming an equatorial metaphase plate with the arms of the chromatids pointed inwards and spindle fiber attaching to the centromere of the chromosomes. Now the spindle fibers attach to the paired chromosomes, and this spindle fiber arises from the centrioles. Now the next phase is the anaphase 1. In the anaphase 1, there is a separation of the chromosomes, and one pair is attached. One pair begin to move this side, and the other pair begin to move to the opposite pole. And these two separated chromatids begin to pull in the opposite poles because of the shortening of the spindle fiber. And the next stage is the phase where the 2 separated chromatids, they begin to reach to the opposite pole, and there is a reappearance of the nuclear membrane. And from one cell, the two daughter nuclei begin to visible inside one cell only. So in this telophase stage, there a single cell contain two daughter nuclei, and the chromosome number, both the nuclei contain the same number of chromosomes. After telophase is followed by cytokinesis. Cytokinesis refers to cell division. So a constriction appears from the middle of the cell, and this constriction deepens till the two nuclei separates completely, and two cells, that is two daughter cells, arise from one cell. So the two daughter cells have the same number of chromosomes containing centrioles and are identical to each other. So this is all about meiosis 1.